Hello my dear students, this is Dr. Pooja, your biochemistry mentor. In this video, we are going to discuss about the metabolism of HDL, which is known as a good cholesterol also. So let's start up with the discussion on metabolism of HDL. Right. Now, what do you mean by this HDL? First of all, this HDL is actually a lipoprotein. Okay. And it is known as heavy or high density lipoprotein. Right. And this is also known as good cholesterol. Because we have studied 4 lipoproteins, 4 or you can say 5 lipoproteins, and in my previous videos also I told you what is the difference between all the 4 lipoproteins. So depending upon the density, depending upon the protein content and the lipid content, these lipoproteins are divided into 4 categories. So HDL is known as a good cholesterol. Why? Because here it is having least lipid content. Right. So obviously if it is having a least lipid content, it will have maximum protein content. So if it has maximum protein content, whenever these lipoproteins are separated on the basis of this electrophoresis, the fastest moving fraction towards anode is this HDL only. So this HDL is also known as lipoprotein alpha. There are other lipoproteins also. This lipoprotein A is there. Most of the students get confused between lipoprotein alpha and A. So HDL is alpha. Why alpha? Because alpha the fastest moving fraction. Right. Followed by, you can say, VLDL, LDL, and then comes the chylomicrons. Right. So this is very important. Then another important point which I have told you is good cholesterol. Now why it is known as a good cholesterol, why LDL is known as a bad cholesterol? The function of HDL is to bring cholesterol from all the other, you can say extrahepatic tissues, and hand it over this cholesterol to the liver. So basically it does not let cholesterol to be deposited on the extrahepatic tissues. So again this is reducing the risk of atherosclerosis. Okay. But now the question is how this HDL helps in this process. So that is what is HDL metabolism all about. So now see, this HDL has specific apoproteins. Obviously I told you, because in my previous videos in which I have discussed about the metabolism of chylomicrons and VLDL, we've been studying the specific apoproteins for them. So HDL ke liye the specific apoproteins are, one is Apo A-I, right, Apo C, Apo D, and Apo E. And the most important, this Apo A-I is the activator for LCAT. Activator for LCAT. Now what is this LCAT? This LCAT is actually, it stands for lecithin, cholesterol, acyltransferase. Lecithin, cholesterol, acyltransferase, it is a very important enzyme in a sense that as you can see from the word itself, it is forming a cholesterol ester. Because always remember, ये जो cholesterol है, this cholesterol obviously we get from the diet, and it is synthesized also in the body. So I can say synthesis or biosynthesis of cholesterol takes place in the body. Now this cholesterol 70% moves in the form of a ester. So this cholesterol, which is actually, if we talk about the structure of cholesterol, it is amphipathic in nature. Right? And this cholesterol will combine with lecithin. Lecithin, which we know it is a phospholipid, right? And it is going to form cholesterol ester and lysolecithin. And if we talk about the enzyme here, the enzyme is LCAT. Lecithin cholesterol acyltransferase. So what happens is it leads to the formation of this cholesterol ester. We will be again discussing about this LCAT enzyme in detail when we are going to discuss about the metabolism of cholesterol. But here you just remember one thing, that LCAT enzyme is responsible for esterification of cholesterol, because HDL metabolism actually deals with cholesterol. Okay, and lysolecithin. Now what is happening is, this HDL comes from, this is the liver, and say this is the intestine. So what will happen is, this HDL will be carrying the cholesterol. Now here what you have to remember is, the cholesterol which is there in this HDL, it is amphipathic. So it is going to give a discoidal shape to HDL. Okay. So I can say that this is the discoidal shape, right? And in this discoidal shape, it is having HDL plus cholesterol. And since cholesterol is amphipathic, the shape is little bit different. Now what is happening is, it has which apoprotein? Apo A-I, as I have mentioned here. You can see here. Apo A-I is activating LCAT, right? What is the function of LCAT? LCAT is leading to the synthesis of the cholesterol ester. That is what exactly happens here. So what will happen is that Apo A-I is activating LCAT. One thing. On the other hand, if we talk about the peripheral organs, say these are the peripheral organs. Here also cholesterol has to be converted into cholesterol ester, plain and simple, because HDL का function क्या है? HDL is going to transport this cholesterol, yeah, it will give this cholesterol to the liver so that the synthesis of bile acids takes place, and there are so many other functions of this cholesterol. So basically HDL is just helping the transport of cholesterol from the extrahepatic tissues to the liver. So what are the enzymes which helps in cholesterol? What are the enzymes which help in the function of HDL? And what are the enzymes which help in actually the transport of cholesterol is what is the metabolism of HDL all about. So in the peripheral organs there are already the enzyme called ACAT, which is acyl cholesterol acyltransferase. There is a difference. Acyl cholesterol acyltransferase is present in the adipose tissue, and LCAT enzyme is present as HDL in the blood vessels. Okay, so this ACAT enzyme, this will also help in the same function like this. And there are three types of proteins which are required: ABCA1, ABCG1 and SRB1. ATP binding cassette protein A1, ATP binding cassette protein G1 and scavenger receptor B1. So they all will help cholesterol to be converted into an ester. The moment cholesterol becomes an ester, it forms a spherical shape like this. So we can call it as spherical HDL. This is very important. So first of all, see how it is going on, that HDL from the liver and the intestines is going to bind with cholesterol. Now since this cholesterol is having a amphipathic structure, it is going to form a discoidal HDL. Now since the transporter is Apo A1, so Apo A1 is going to activate LCAT, right? That means this cholesterol will be converted into cholesterol ester. The moment cholesterol ester is produced, the shape only of HDL changes. From discoidal, it becomes spherical. On the other hand, there are other proteins also which are helping in this. So ABCA1, ABCG1 and SRB1, they will help in bringing more and more of cholesterol esters. So these cholesterol esters will form a spherical HDL. Now what happens is that the concentration of this will keep on increasing. Okay, so this HDL3 will swell up. Why it will swell up? Because of continuous, you can say, triggering of this discoidal conversion into spherical HDL. So what will happen is it will immediately form another spherical HD2, or you can say HDL2, whatever you can write. HDL2 also you can write here. Spherical HDL2. Okay. So now what will happen is that whatever amount of cholesterol is there here, this will be accepted by a SRB transporter here. So SRB transporter will accept cholesterol esters and it will send them to the liver and it will be converted back to this. Again LCAT, and this will give to this spherical HDL again. So like this the chain keeps on going, and this is your HDL cycle. Okay, so now why we call this as reverse cholesterol transport? Because यहाँ पे everything is going on in a reverse manner. Okay, that is why we always say that HDL is responsible for reverse cholesterol transport. For reverse cholesterol transport. Very important, right? Now, in simplest language, we can say why? Because it brings cholesterol from the extrahepatic tissues to the liver. And what is the function of cholesterol? Because in the liver, cholesterol can undergo bile synthesis. That is why it is a good cholesterol also. Very, very important. Clear? So this is what is the metabolism of HDL. Now we have to jot down in points. So I can simply say that HDL metabolism starts from the liver and intestines. Right. So what happens is it combines with cholesterol. Combines with cholesterol. Right. And cholesterol is amphipathic. This is the most important point to remember. Cholesterol which is amphipathic to form a discoidal HDL. Right. Now, we have already studied about the apoproteins. So, Apo A-I, which is activating LCAT, right, will make cholesterol esters. Cholesterol esters. Right. On the other hand, other peripheral organs are also having different proteins like ABC-A1, which is ATP cassette binding protein A1, right. Then again ATP cassette binding protein G1, and the third one is scavenger receptor binding protein B1. So these are the ones, they will again be accelerated by LCAT and they will form cholesterol esters. Right. Now ester, obviously the structure will become different. So we can say that the structure will now be known as, instead of discoidal we get spherical HDL. So HDL becomes spherical. Becomes spherical. Why spherical? Because of the formation of cholesterol esters. Now this HDL is also known as spherical HDL, or we can call it simply as, you can say HDL3, like that. Up it will keep on accepting, so HDL swells up. HDL swells up and is converted into, say I will write just spherical, so HDL3 I can write, and is converted into HDL2, because it is accepting. Now what will happen is this HDL2, again it is spherical only, the structure will be spherical. Now this HDL2 will give the cholesterol to the liver. So HDL2 will transfer cholesterol esters to the liver with the help of SRB1 transporter, and itself will be converted back to HDL3. Right. So this is what? This is HDL cycle. So let's just see quickly the diagram. So here you can see that if we talk about HDL plus cholesterol, this is discoidal shape. LCAT enzyme and ACAT enzyme are forming spherical, which will be converted into this because it will increase and it will be like this. This is HDL cycle and this will be accepted by SRB transporter. So this is HDL metabolism. Now there are other lipoproteins also which are somewhat, they are resembling this. जैसे most of the students get confused between this. जैसे HDL is known as lipoprotein alpha. Just keep this thing in mind. Lipoprotein alpha is HDL. Why HDL? Because it is the fastest moving fraction. Very important. Right. There is one more lipoprotein which is known as lipoprotein A or LPA. There is a difference. Now what is this lipoprotein A? It is a plasminogen analog. Basically this LPA is a plasminogen analog. And it is accepted by Apo B100, which is an acceptor of LDL by a disulfide bond. Right. So what happens here is that it is an plasminogen analog. So what will happen is plasminogen, which has to be converted into plasmin due to the presence of this LPA, not alpha, LPA, it will inhibit the clot lysis, thereby increasing the risk of thrombosis. Very important, right? So this is what? This is a lipoprotein A and this is lipoprotein alpha. One more is there which is known as lipoprotein X. Now what is this lipoprotein X? What happens? Basically, whenever there is obstruction in the flow of bile duct, obstruction in the bile duct, it will lead to increased concentration of cholesterol in the liver. Right. So what happens is when this cholesterol concentration increases in the liver, it binds with or it combines with phospholipids, forming, plus I would write, forming this LPX. And this LPX becomes an indicator of cholestasis. Very important. Okay. So, these are, I won't say the lipoproteins, but they are resembling. They are like the similes. Most of the students get confused between this. So, always remember beta, there are only 4 or 5 major lipoproteins. We can call them as alpha, pre-beta, broad beta, beta and chylomicrons. But lipoprotein A is altogether different, which is actually a harmful one also, and LPX or lipoprotein X. So overall, if we talk about the apoproteins or lipoproteins, we saw that these apoproteins, when they are combining with the lipid fraction, they are forming the lipoproteins. And what are the different type of apoproteins? If I talk about Apo A1, Apo A1 always activates LCAT. Right. Apo A2 also activates LCAT. But Apo A3 inhibits LCAT. Because wherever there is an activator, there has to be an inhibitor also present. So it will inhibit LCAT. Then if we talk about Apo C1 or C2, so Apo C2, we know it always activates LPL. This also is very, very important. Okay. Then Apo B48 is specific for chylomicrons. And Apo B100 is specific for VLDL and LDL. And why we have named them as Apo B48 and Apo B100? Because they are the only two apoproteins I would say. Rest you see, in all the apoproteins I have not given any number. A1 hai, A2 hai, A3 hai, C1 hai, C2 hai, but here we are writing 48 or 100. So only two apoproteins which are synthesized by a process which is known as RNA editing or RNA processing. Right? So this is how we study the metabolism of the lipoproteins. HDL known as good cholesterol, LDL known as bad cholesterol. And if we have to differentiate, I can simply say that, suppose say this is the liver and these are the extrahepatic tissues. Right? What is the function of LDL? LDL will take cholesterol and deposit. Right? Whereas HDL will do the opposite process. HDL will bring a cholesterol to the liver. Okay? Because always remember, cholesterol is required for the synthesis of bile acids here. Right? And that is why we refer them to as good cholesterol and bad cholesterol. So this is all about the metabolism of HDL, and that is why we can say that this HDL is because it is doing the reverse or the opposite. So that is why we say HDL is responsible for a reverse cholesterol transport. This is very, very important. Right. So this is all about in this video on the metabolism of HDL, which is a friendly cholesterol also. I hope this is clear to all of you. So thank you so much.